I don't know if I can send it to you.
involves both facilitated diffusion and active transport mechanisms. You're not going to have to memorize the steps of an action potential. But through the lens of Unit 2, why is the neuron relevant? The neuron is relevant because you have good. So number one, it does have high surface area to volume ratio, so it's very efficient at diffusion, which is important because the neuron is relying on diffusion to communicate with other cells. And this communication with other cells is going to help regulate your physiology. So let's look at protein A. Protein A could be like the sodium-potassium pump. So primary active transport, which means the pump is being phosphorylated to move 3 sodium out and 2 potassium in. The potassium that is brought in is allowed to passively diffuse back out. So I'll use a dash arrow or dash line for facilitated diffusion, so high to low. The solid line for active transport, low to high. That creates a voltage. You need to know what a voltage is. You need to know what an electrochemical gradient is. Voltage and electrochemical gradient, it's the same thing. So the sodium is high on the outside and low on the inside. So there's a chemical gradient that is, you can imagine, think of it like a pushing force that's pushing sodium from a higher concentrated region to a lower concentrated region. But then there's also, on this side of the neuron, on the cytoplasm side, there's a negative charge. So that negative charge attracts the sodium. So you can think of that as like a pulling force. So there's a pushing force, the gradient, the chemical gradient, and then there's a pulling force, that's the electro part of an electrochemical gradient. And that just means that sodium will rapidly rush in if given the opportunity. So during if you guys are gonna clown around, I get to ask you to leave. If a neuron is stimulated, sodium channels open, and you just need to know that sodium generally wants to move into a cell. And the reason it wants to move into a cell is because all cells have the sodium-potassium pump. When sodium rushes in, the neuron depolarizes. In other words, the inside of the neuron within this region temporarily becomes more positive and the outside becomes negative. At a certain point, those sodium channels are going to close and potassium channels are going to open. You need to know that generally potassium wants to diffuse out of a cell because it's pumped in thanks to the sodium-potassium pump. Notice that potassium is also diffusing down its electrochemical gradient. There's a pushing force pushing it from high to low. There's also a pulling force on the outside pulling that potassium out. That causes the inside of the cell to repolarize, and that is the action potential. So, I mean, if you see a neuron on your exam, it'll be through the context of understanding what primary active transport is, what facilitated diffusion is, what is an electrochemical gradient, and like what role does that electrochemical gradient play in the movement of ions across a membrane. And then also I could say, hey, when the action potential gets to the end of the neuron, what happens? Exocytosis, right? So you could get a question about, well, you know, what is exocytosis? What happens as a result of exocytosis? What do we find in these vesicles? Typically you find molecules that are too large to cross the membrane via facilitated diffusion or active transport. That's why exocytosis is required to get them out of the cell. I could say, hey, let's expose the cell to a toxin that blocks the sodium channels. What's going to happen? Well, sodium can't get in. which means the depolarization can't happen, which means the neuron can't fire. Or what if I mutate the sodium-potassium pump and make it less functional? Well, then the neuron is going to have a problem generating a voltage. So things of that nature. Does that cover the neuron for you, Aaron? Okay. What else? Yeah.
Secondary active transport.
Secondary active transport. So secondary active transport. Where's my eraser? Does anybody see my cloth for erasing? That's a problem. Okay. Oh well. All right, in secondary active transport, here's a cell. It's going to use, I'll say protein A again, the sodium-potassium pump. So it's going to pump sodium out, and it's going to pump potassium in. This is helpful because it helps generate a voltage across the membrane. And that voltage, you can think of it as just stored potential energy. It's basically turning the membrane into a battery. There's another protein over here, you can think of it like a little wheel. And this protein it's not a channel, right? So in the neuron, it's a channel. So as the sodium rushes in, that sodium isn't being used to power anything. But in this case, this protein that's shaped like a wheel can allow sodium to rush back in down its electrochemical gradient, and that can cause the wheel to spin. And as the wheel spins, work can be done. And so one example of the type of work that could be happening here is glucose, let's say, has a low concentration out of the cell and a high concentration inside the cell, but the cell wants as much glucose as it can possibly get. The cell cannot rely on diffusion and wait for glucose to come in. In fact, if it puts GLUT on its membrane, which is a protein that does facilitated diffusion, if it puts GLUT on its membrane to try to get the glucose out here, it's actually going to lose glucose because glucose will move from high to low, I guess dashed for passive. So that would be counterproductive. So instead what the cell can do is it could use a protein like this to spin, and as it spins, the protein grabs onto glucose on the outside and tosses it to the inside. So even though this is a passive process, the sodium diffusing down its electrochemical gradient is passive, we would still call this overall mechanism secondary active transport because it's using an ion gradient to power the pumping of something else. And in this case, they're both going in the same direction, but the direction is not important. What's important is the gradient. Here we're going from high on the outside to low on the inside, but for glucose, we're going from low on the outside to high on the inside. So glucose is moving against the gradient while sodium is moving down the gradient. That's secondary active transport. Does that clarify?
Yeah.
Okay. What other questions do you guys have? Or what other topic do you want to brush up on?
Cell death?
Cell death. Okay. Does no one see my eraser? No one sees mine. The way that you might encounter cell death on the test is through a question that talks about the lysosome. So a cell could get a signal to die. So we watched a video of a killer T cell with a certain protein sticking out, binding to a receptor on a cell that is ultimately going to die. And so as a part of that interaction, lysosomes will fuse to organelles throughout the cell so that those organelles can be broken down. Aaron, who asked the question? Oh yeah. So that's all you need to know about. We're going to get deeper into cell death in Unit 4, but for now, if a cell is instructed to die, it will use its own lysosomes to break down pieces of itself. And cells will break down pieces of themselves all the time as a regular maintenance process. But if we're talking about like the destruction of the nucleus, the destruction of the ER, the destruction of all of the organelles, it's because the cell is preparing itself for a quiet, controlled death. So you just need to know, well, like, what is the role of the lysosome in that? The lysosome contains hydrolytic enzymes that can hydrolyze larger macromolecules into their building blocks. That's it. The only other thing I can think of as far as the lysosome goes is we know that the pH of the lysosome is very low. And the reason for that is because protons are pumped into the lysosome. So there's a possible active transport component to any kind of lysosome question. I could say, like, what if an individual has a disorder where their proton pumps don't work? What's that going to do to the effectiveness of the lysosome? It's going to decrease it. And now this individual has problems digesting, not digesting food, but the cells that make up the individual have issues digesting things that they've taken in through endocytosis, or the cell has problems recycling its own organelles, and that could be problematic. Yes? Okay. What else? What other topic? Say,
osmosis, but not like the calculation.
Yeah, just like general
Yeah.
osmosis stuff? Okay. So, what you need to remember about osmosis is that when I have a semi-permeable membrane, like a phospholipid bilayer, there's a solution on one side and there's a solution on the other side. From a cell's perspective, the solution on one side is called the cytoplasm, and the solution on the other side is just the extracellular solution that bathes the cell. So if I have solutes that are trapped on one side of the membrane in one solution, and solutes that are trapped on the other side, normally these solutes would diffuse down their own concentration gradient, and I would see the net movement of solutes going from the left into the right. But if the solutes are trapped, water instead will balance the solute concentrations out. Let's say the solute concentration over here is 0.4 molar, and the solute concentration over here is 0.2 molar, right? It's less concentrated. Osmosis is just the movement of water into the more concentrated solution in order to dilute it. So this solution becomes concentrated as water diffuses out of it, and this solution becomes diluted. That means this is going to decrease, this is going to increase, and they're going to meet somewhere in between. Once the molar concentrations are balanced out, we would say that the system is in osmotic equilibrium. Or, I mean, you have to remember, for a plant cell, a plant can reach osmotic equilibrium without balancing the concentrations. They can reach osmotic equilibrium through pressure instead, but we can get to that if you want to and if we have time. Remember, this is not active transport. I've had interactions with students that seem confused because I'm drawing an arrow from the less concentrated side to the more concentrated side. But I'm not talking about the movement of solutes. I'm talking about the movement of water. This side has more water because it has less solutes. Remember, when I'm talking about the movement of water, I'm talking about the movement of free water. Water that is locked up around the solute cannot move. And so you can think of solutes as like water traps. They're trapping the water. So this side, there's more water trapped because there's more solute. So this side has more free water, this side has less free water, and so water is diffusing down its own concentration gradient. This is not active transport. Water is not actively transported across a membrane. The only thing that can be actively transported across the membrane are solutes, and then water can follow passively towards the higher concentration solution. But water is not, this is not active transport, okay, of water. We would call the side that is more concentrated with solute hypertonic. So the hypertonic solution will always have water coming into it, and the hypotonic solution will always have water leaving it. So where the water goes just depends on which side is hypertonic versus which side is hypotonic. If I say that the cytoplasm is hyper, well, now you know that the cell is going to swell as water enters. Is it going to rupture? I don't know. It depends on how much it swells. It depends on how much water it takes to enter the cytoplasm before reaching osmotic equilibrium. If it's just a little bit of water, then the cell's probably okay. But if the gradient, if the solute gradient is extreme, and remember, when we're talking about osmosis, I really should have written these not as molar, but as osmolar, because you have to convert molarity to osmolarity. If inside the cell, it's 0.3 molar, and, well, let's make it more extreme. Like inside the cell, it's 0.8 molar, but outside, it's 0.1 molar. That's a lot of water that has to rush in to dilute this down to 0.1, because remember, in this scenario, the cytoplasm is changing much quicker than the extracellular solution, because the extracellular solution is large in volume. The cytoplasm is not large in volume. So can this cell handle enough water to get down to 0.81, or is that just too much water? Is it going to explode? I don't know. You would have to have more information to know whether or not the cell is lysing. Like, think back to the aquaporin data that we did near the beginning of class, beginning of the unit. If I'm measuring the cell volume and suddenly there's no more volume to measure, well, chances are it's because the cell has popped. If this is a plant cell, then I don't really need to think about the cell popping. That's not going to happen. Instead, what's going to happen is the cell wall is going to start pushing up against the membrane, generating a pressure, and that pressure could prevent the cell from overexpanding and popping. And so, like, how does pressure apply here? Like, without getting too deep into the math, because there's not that, we don't go that deep into the math in this test. I might say, hey, the water potential out here is based off of this. I'm not going to actually do the calculation, but you could do negative ICRT, and let's say the water potential out here comes out to be, I don't know, -8. And let's say the solute potential here is negative 15. And the cell starts swelling and pressure starts becoming a factor. How much pressure is going to be generated to get this cell to a water potential of -8? Okay? For an animal cell, we don't factor this in, because remember, animal cells don't have a cell wall. Okay. Saeed, does that cover osmosis for you?
Yes.
Okay. What else? Here's my
Well, step it out. Ask a question.
Step
Step it up.
This
This is why you're here. For the good of the group.
Why would I do something
All right, yes.
stupid?
Okay. Yes. Yes. All right, endo and exocytosis.
Let us down.
Why not?
Okay, so here's the cell. Let's say the cell wants a large protein, protein X. It's a large protein, so it's way too big to cross the cell by facilitated diffusion or active transport. Because remember, when we're talking about things crossing the cell through transport proteins, the range of size ranges from a single proton, which is as small as you can get, to a monomer or a monomer slash a steroid, which is about the same size as a monomer. Anything bigger than that requires either endocytosis or exocytosis. So if protein X needs to get into the cell, that cell can fold its membrane inward and then pinch the bubble off of the membrane and form a vesicle. And now what happens just depends on what the purpose of protein X is. Maybe the cell wants to break down protein X and use those amino acids to build its own proteins. So what organelle does it need to fuse it with? A lysosome, to break down protein X and use those amino acids for other proteins it needs. Or maybe protein X is an important protein for mitochondrial function. So maybe this vesicle now fuses with the mitochondrion and dumps protein X into the mitochondrion. It just depends. So there's three ways that cells can do endocytosis. They could do phagocytosis, which is sort of, think of it as cellular eating. So this cell can eat a smaller cell, like a bacteria cell. That is going to involve fusing with a lysosome. Mostly, like rarely, the smaller cell will live inside of the bigger cell. That doesn't happen very often, but there was an important period of time where that did happen. And right, that led to the evolution of the mitochondria and also the evolution of the chloroplast. But most of the time it's going to result in the destruction of the smaller cell. There's penocytosis. Penocytosis is, you can think of as gulping. Basically the membrane is just, it looks like under a microscope, randomly pinching inward so that lots of solutes can be taken in at once. So rather than like solutes individually moving through transport proteins, the cell is just gulping a whole bunch of solutes all at once. And then there's receptor mediated. I'll just say receptor endocytosis. So like in this example of protein X, the cell maybe has a receptor that binds to protein X, and that's telling the cell to bring it in. That way the cell is very specific in terms of what it's bringing in. Penocytosis is not very specific, it's just gulping. Phagocytosis is not very specific, but receptor-mediated endocytosis is. And then the opposite of that is exocytosis. And so here you have vesicles that contain, I don't know, like a neurotransmitter, for example, or like insulin, like some sort of hormone. And when we looked at a beta cell, we saw that a beta cell can depolarize to release insulin. And so when a beta cell depolarizes, that depolarization triggers exocytosis. So the vesicle fuses to the membrane, and whatever material is within that vesicle is now released into the extracellular environment. Endo versus exocytosis. Yeah.
You need to know all the endocytosis